young minds with this scientific knowledge base, promote science and technology, and demonstrate how science could lead India towards a developed and self-reliant nation. As a part of this unique yatra, today, Regional Center for Biotechnology proudly showcases its world-class infrastructure and excellent research base. Sorry for this interruption. Um, so, Regional Center for Biotechnology proudly showcases its world-class infrastructure, excellent knowledge base through this 2-hour journey in an attempt to excite youngsters joining us today and motivate them towards a career in science, research, and entrepreneurship. At the outset, I request our Honorable Executive Director, Professor Sudhanshu Vrati, who is an inspiration behind all of us in our journey to achieve excellence and perfection in our initiatives, to deliver the inaugural address and formally welcome the participants. Professor Vrati, please.
Thank you, Deepika, and a very good morning to all those who are joining us this morning to be part of the Vigyaan Yatra hosted by the Regional Center for Biotechnology, or the RCB. I welcome you all to be part of this yatra. RCB is an institution of national importance established by the Department of Biotechnology, Government of India, under the auspices of UNESCO. This Vigyaan Yatra we bring to you as part of the India International Science Festival 2020 with talks from our young scientists showcasing the RCB research and its infrastructure. IISF is an annual event organized jointly by Ministry of Science, Ministry of Health and Family Welfare, and Ministry of Earth Sciences of the Government of India with the Vigyaan Bharati, with support from a large number of institutions. IISF organized to display India's contribution in the field of science and technology and to motivate young scientists to find solutions to the burning issues of our society. A long-term objective of the festival is to encourage young minds to study and work in scientific fields. IISF also shows the way how science, technology, engineering, and mathematics, that is STEM, could provide us with the solutions to improve our lives. I hope you will be able to participate in the IISF this year, which is being organized from 22nd December on a virtual platform. This will be the largest science festival to be hosted on a virtual platform. The central theme of this year's festival is Science for Self-Reliant India and Global Welfare. For now, please enjoy the scientific talks by RCB and a movie or couple of movies displaying the RCB's infrastructure. So welcome again, and thank you for joining us.
Thank you, sir, for encouraging us to connect with the students and the society through this unique outreach initiative. We will now walk you through an overview of Regional Center for Biotechnology through a short video that showcases our uniqueness and will acquaint you with our strengths, so that if you are interested in any of these areas, RCB is the place to look out for in future. We start the video, please.
Regional Center for Biotechnology is located in a tranquil environment along the Gurugram-Faridabad Expressway. It is an autonomous institute established under auspices of UNESCO by the Department of Biotechnology, Government of India. The primary focus of RCB is to provide world-class education, training to create high-quality human resource in biotechnology. and conduct innovative research at the interface of multiple disciplines. The infrastructure at RCB has developed at a rapid pace since inception of the institution in 2010, with state-of-the-art research labs, teaching labs, innovative classrooms, central instrumentation facilities, small animal facility, biosafety level 3 facility, a biorepository and state-of-the-art advanced instrumentation facilities like the Advanced Technology Platform Center and a bio-incubator which serves as a reservoir for incubating ideas in the area and help them reach commercialization. There are various areas of research in RCB. The research group under the area titled Cell Biology, Development and Behavior focus on unlocking the secrets of cells, how they divide and how stem cells develop into muscle, in addition to studying the cellular and molecular origins of how organisms behave. Areas of agricultural biotechnology aim to tackle some issues by harnessing the existing genetic diversity in plants and their inherent capacities to adapt to abiotic and biotic stress conditions in order to develop innovative and durable methods of crop improvement. Groups working on infectious diseases, both bacterial and viral, aim at developing novel therapeutic strategies against infectious agents using tools such as mass spectrometry and microscopy. These researchers will shed light on cellular and molecular bases of bacterial infection and the response of the human host towards these infections. They are trying to find novel solutions to protect the people against diseases such as chikungunya virus, Japanese encephalitis virus and dengue virus. Protein structure and design groups are structural biologists who have a molecular view of life and use cutting-edge methods like macromolecular X-ray crystallography or cryo-electron microscopy to imagine biological molecules in their functional state. This information is used to develop novel therapeutic strategies against pathogenic bacteria and viruses, and for protein engineering to prepare molecules with desired properties. Researchers at RCB are also working towards developing innovative biotechnological applications such as novel drug delivery systems, new diagnostic tools, novel engineered protein with improved desired proteins, and improved methods to obtain desired products in large scale. These researchers subject laboratory and clinical samples to cutting-edge microscopy, proteomics and genomics methods coupled with flow cytometry to shed light on the etiology of disease and utilize this knowledge to identify solutions to these disorders. The quality of research conducted at RCB is excellent, with faculty winning top honors like the Shanti Swaroop Bhatnagar, the National Bioscience, the Innovative Young Biotechnologist Awards, and the Ramanujan Ramalinga Swamy and DBT Welcome Alliance Fellowships. Since research-based learning is the hallmark of RCB, the academic programs here are deeply enmeshed with the research programs. The academic programs fulfill RCB's core mandate to provide quality education and training in the area of life sciences and biotechnology. The PhD program in biotechnology is for students who are interested in working at the interface of multiple disciplines to find novel solutions for problems in health and agriculture. RCB has recently started integrated PhD program in biotechnology, offered to students with graduate degree in any discipline of science. PhD program in biotechnology is for students who are interested in working at the interface of multiple disciplines to find novel solutions for problems in health and agriculture. RCB has recently started integrated PhD program in biotechnology, offered to students with graduate degree in any discipline of science from India and abroad. The program provides extensive learning opportunities in the broad field of life sciences and biotechnology. RCB has also recently initiated an interdisciplinary doctoral programs in the area of biostatistics and bioinformatics through collaboration with the global pharmaceutical giant GlaxoSmithKline (GSK), being conducted in partnership with other institutions by creating a virtual faculty pool. The focus of this program is on creating specialized manpower for the healthcare industry. As an outreach activity, RCB also offers research training programs for research-driven undergraduate and postgraduate students of science from various universities. Trainees get a realistic experience of several facets of modern biological research that sets the tone of their embarking on a research career. Throughout the year, RCB hosts and organizes regular academic events like the RCB Bioimaging School, national and international conferences, seminars, symposia, and training in the frontier area of basic and applied sciences in topics such as infectious diseases, drug discovery, etc., to disseminate advanced knowledge, exchange ideas, foster national and international collaborations, student exchange, and networking opportunities. RCB also holds scientific communication and communication workshops for the benefit of young scientists in India. RCB has a fully functional library and houses 500 scientific textbooks and 100 administrative books in multiple copies. In addition, an electronic library provides access to a vast range of primary literature in the form of peer-reviewed journals and reviews. RCB offers faculty residences and excellent students' facilities, including on-campus air-conditioned hostel accommodation, modern library, meeting rooms, seminar rooms, and auditorium. The hostel and student facilities are conveniently located and only a short walk away from the classrooms and laboratories. In addition, the campus also has sports and recreation facilities which encourages all-round development of the students. Campus also provides a childcare facility for all students and staff to help them continue their studies and work while their babies are being taken care of. Kridangan, the creche housed in the faculty building, is an asset on campus for employees of NCR Biotech Cluster. The spacious cafeteria in the campus serves hygienic, nutritious and delicious meals and beverages at reasonable cost. RCB also contributes towards creating resource for researchers from all over India and has engaged with institutions such as the European Synchrotron Radiation Facility in France towards this end. RCB is also involved with the UNESCO towards developing policy to ensure sustainable development at the global level and specifically in the Asian region. RCB is a young institute which is growing day by day with excellent opportunities for young people to make a career in the area of biotechnology.
That was a brief overview of RCB. For the next segment, we will move to the various research domains, exposure to high-end research facilities at ATPC, and its connection to applied aspects of research through our bioincubator. So next, we will handhold you towards the 6 exciting research domains of biotechnology that RCB specializes in, with the help of 6 of our young scientists, each of whom will take you from the basics of the domain to what research questions our scientists are trying to answer in that area. We shall start our journey of motivation towards science with the talk of Dr. Prem Kaushal, who will briefly acquaint you with careers in science, after which he will talk about his core area, structural biology, an area where RCB has hugely contributed towards its basic principle and also towards the latest by things like screening the drug molecules for the novel coronavirus. Over to Dr. Prem Kaushal, please.
Thank you, Dr. Bhaskar. Good morning to all Vigyaan Yatri. I will start with conveying the message that scientific research is important and it is the best career option, as Dr. Bhaskar already mentioned. See, the research carried out in science and in the field of science and technology, that is the scientific research, brings innovation and invention to our society. And that has helped the life, the quality of life we live, and it has changed forever. So I will give you few examples to start with. One example is very old example. You see, the Wright brother first time invented the mechanism of flying, how object can be flight. And now you see a Boeing 777 that it's empty when this plane is empty. Its weight is 134,000 kilos, and it flies 40,000 above the ground over the cloud, and it is possible only because of science and technology. On other hand, if a leaf detached from a plant, it falls down, it cannot stay in air because of gravity. So this big vessel is carrying above the cloud. Is it not amazing? And other discoveries that has improved the quality of life, like discovery of penicillin, first antibiotic, saved thousands, millions of lives. You see, the polio vaccine has improved the quality of life. Otherwise, there would have been so many people paralyzed because of polio. The cholera vaccine, the first commercial vaccine. The treatment for the TB, there is almost cure for the TB, has saved again a lot of lives. X-ray has improved the quality of diagnosis. Without looking into the body, we can see the inside the structure of the things, whether they are fine or good, bad. MRI has improved the diagnostic. You see, the computer and the internet, it has changed. Although in this terrible pandemic phase, we cannot move around freely, but still we are connected. We are sitting here. Some of you are sitting all over the India, maybe abroad also, but still we are connecting. All these things are because of science and technology that brought the new invention, innovation. India also has made huge progress through science and technology. I have picked only one example because of time constraint. You see, the green revolution of India is the one of the best example. When India got independence, before that in 1942, India witnessed a great famine of Bengal. People died because of hunger. There was a food shortage. And over the time, you see in this graph, over 40 years, India produced 7 times more wheat that ever produced. And M.S. Swaminathan, the father of Green Revolution, was the key player. And now our population after independence, our population is 100 crore more. Still, we have surplus amount of food. All is because of science and technology. Now, the latest thing is the corona. For the corona pandemic, there are 2 vaccines available in the market. Both are mRNA vaccine. Basically, this is the mRNA of this spike protein that is chemically synthesized and, and, and put it in a nanoparticle, that's a lipid molecule, and transferred to the cell. Now, cell will know that it's its own mRNA. It will translate and make this spike protein. And once this spike protein is in our body, our immune system will produce antibodies, and there will be antibody already in our system before virus attacks. So first, the vaccine was developed by Pfizer, was administered on December 8th, and just 3 days back on December 18th, second vaccine for coronavirus developed by Moderna also get FDA approval. To understand this, I will take you a little bit in the history. This is the Watson and Crick at Cavendish Laboratory in 1951. They demonstrated that the genetic information is encoded in double-stranded DNA. Crick is explaining, and it is a double-stranded, and found out how the structure of DNA looks like, and gave the interpretation how genetic information will be translated to protein. And that gives rise to the work of molecular biology and the understanding of central dogma. Central dogma is the information encoded in DNA is transcribed into the mRNA. It is possible by reverse transcription, it can go back also. By replication, it can synthesize another strand of DNA, and that mRNA is translated into the protein. Now, this is the protein that actually functions. But if now people over the 60 years of understanding, people have now chemically synthesized this RNA molecule and added to the cell and synthesized this protein. So it is basically by this deep understanding, we are now able to fool our body that this mRNA is your own and make it protein. And this mRNA vaccine is a fruit of 60 years of molecular biology understanding that gives more than more than 90% effective vaccine. It's a record. Okay. Now it comes why the research is the best career option, and one of the best thing is you have to choose. You have the freedom to choose your direction. You have the freedom to choose the research topic what you like. And this is the only career where you have the option in what you really want to do. Obviously, you have to do a meaningful work, and there is no other career where you have the option what you really want to do. And second is, you will be, if you are a researcher, you will be participating in great era of discovery. Like this time, people involved in finding the ways and cure for COVID, they are involved in a great era of discovery. And as a researcher, you will be being a part of international community joined by a common interest. And another reason is you will have a social and youthful job. Means it doesn't matter how old you grow, your lab will be full with young minds, postdocs, PhD students, and always a youthful life. This is a big thing, that you will be always doing good for the society, always thinking of the societal problems. And there are a lot of fellowships and scholarships involved in research. Means for learning, you are paid. With this, I'm sure you are convinced that scientific research is a great thing and a great option. Now I will take you to the structural biology activities, research in the structural biology field that is happening at our regional center. At regional center, the 4 PIs are conducting research in the structural biology: Professor Deepak Nair, Dr. Venkateshan, Dr. Deepti Jain, and myself. If you see much of scientific observation, we believe much of scientific observation we make by seeing. And this is the first time when Anton Levin book discovered microscope, he introduced a new world, that is microscopic world. And that world was never seen before. And now, immediate after, lot of new things were seen, lot of new discoveries were made. But immediate after, people realized that a light microscope, where the light is used as a radiation source, cannot be able to see the atom of a molecule. And because the wavelength of the light is in nanometer range, and we can see object only of the nanometer size. If we want to see the atom of a molecule, we need a radiation that has of wavelength of angstrom size. And over the time, two of the techniques developed. One is X-ray crystallography, where you have an X-ray beam that has a wavelength of angstrom size. His X-ray beam is shooted to the crystal, and you get a diffraction pattern. And from diffraction pattern, by adding the phase, you get the structure of a molecule, and you can see each and every atom. Another technique was developed was the cryo-electron microscopy. You have an electron beam, and you take a frozen sample, and you get a 2D projection. From 2D projection, you make the 3D projection. With these two tools, we can see each and every atom in a molecule. And now question comes, why we need to see the atom of a molecule? Because you know that all the chemical reaction, either bond formation, breakage, or if a inhibitor is if a drug is inhibiting any molecule, it is happening at the level of atom. These atoms are involved. So that's why it is important to see the atoms. And with these two techniques, people are able to see each and every atom inside a big giant virus like Zika virus, a big mega-Dalton complex such as ribosome complex. At RCB, we are using mainly these two techniques along with other molecular biology techniques to address different aspects of different scientific questions. These are our tools. This is a 120 kVA electron microscope used for negative staining and used to see the object. But we have a very sophisticated equipment that is cryo-electron microscope, where you can see a molecule in its native form, means it is active, and you can trace see each and every atom in the molecule. It is in ATPC. We have two X-ray diffraction system. We grieve the crystal and shoot it here, get diffraction pattern, and get the structural information at atomic level. We use robots to set up the crystallization. And this is another equipment for making grids for cryo preparation. So we have to preserve that in its native condition. This equipment is used for that purpose. Now coming to the different activities different PI is doing. Professor Nair is working on molecular determinants of genomic integrity and plasticity. You see, this is the DNA molecule that I already mentioned. It has two strands. One strand be inherent from mother, another strand be inherent from father. And we need to maintain the genomic integrity so that human beings' offspring look like human beings, chimpanzee look like a chimpanzee. And other hand, there should be some change that is encoded in GC letters here. So we evolve. That is called plasticity. And that plasticity is important because of plasticity or evolution. We evolved from chimpanzees to human beings. Otherwise, even today we would have been like chimpanzees. Professor Nair is looking into on microbes, and if there is a condition, it's a normal condition, what bacteria does, they maintain the integrity. So there is no change in the base pair or mutation. But when there is an antimicrobial agent to escape from the antimicrobial or for stress condition, it uses its plasticity, means it induces some mutation and escapes from these antimicrobial agents. So area of interest are the molecules and molecular pathway that maintain integrity of genome and genomic plasticity to facilitate evolution. Structure-based drug design to develop novel therapeutics against virus, bacteria, virus like coronavirus, etc. And the current activities are going on, the structural and mechanistic insight regarding role of DNA polymerase in DNA replication and evolution, structural mechanism of action of viral RNA-dependent RNA polymerase, structural mechanism of repair enzyme in bacteria and viruses. Another exciting area Dr. Bangdeshan's group is taking is the structural biology of host microbe interaction, bacterial pili and biofilm. That is, if you say this bacteria, and these bacteria, they are our friends and enemies. They does a lot of good thing for us and bad thing also. And some of bacteria, they attach to the host surface by these fine structures, the pili. And Dr. Bangdeshan's group is interested to pinpoint each and every atomic detail, how this interaction with the host takes place. If these are used, these are our friends, these are the friendly mycobacteria, the point will be to enhance this attachment. If they are enemies causing diseases, idea is to block their attachment. So here is the overall research interest in Dr. Bangdeshan's group, visualizing pili and host microbial interface at the atomic level through structural biology tool to understand the mechanism by which microbe adhere to and interact with host surface for colonization and biofilm formation. The current activities are going on to understanding the structural basis of bacterial pilus biogenesis, how that fine structure biogenesis, understanding mechanism by which bacteria attach to the host surface for colonization and biofilm formation, understanding how adhesion strategy varies among pathogen and bacterial beneficial bacterial strains, targeting pili-mediated attachment for combating microbial infection and improving health. Another exciting area is transcription regulation and structure and mechanism. Transcription means when DNA is synthesizing mRNA, at that level, how the things are controlled. Dr. Dipti Jain's group is working on a very exciting area. That is, these bacteria, they have flagella, and flagella is they use for locomotion to find such food and move here and there and colonize to other parts. And then they also have a strategy that if there is enough food and things are normal, they have these flagella and they walk around and such. Then there is the stress of shortage food, ATP less. They form these biofilm. They stay in group. But when they need a flagella, when they don't need, it is regulated by a complex machinery at transcription level. And Dr. Jain's group is looking on this aspect, and the area of interest are the transcription regulation of flagellar and biofilm gene network in Pseudomonas aeruginosus. This is a pathogenic bacteria. Allosteric mechanism utilized by transcription factor responsive towards small metabolites. And the current activities are going, current questions are addressed are what is the structural mechanism of biofilm formation in Pseudomonas aeruginus? what is the structure mechanism of antimicrobial resistance in Pseudomonas and Staphylococcus aureus. Both are pathogens. Now in my group, I'm looking into the structural aspect of translation regulation and ribosome biogenesis. You see this complex machinery, this is ribosome, and it synthesizes all the protein in our body, including the hemoglobin that you can see the transport oxygen from lung to tissues. And this machinery is made up of some 50 proteins, RNAs, and our group is interested to see how this machine exactly assembles inside the cell. Another thing is this Mycobacterium tuberculosis that causes TB. When it enters into our body, goes to the macrophage. Most of time, in 1/3 population, world population, it goes in dormant state. Dormant state is like similar, the hibernating state that reptile goes when there is unfavorable condition. And same in when your human body is strong, it goes in dormant phase. When your immune system weakens, it reactivates and causes the disease. And we are looking here how it synthesizes protein in this dormant or hibernating state. So the overall area is to unravel the structural basis of functioning of macromolecular complexes involved in translation regulation and ribosome biogenesis, thereby to identify the potential drug targets. The current activities are to understand the translational strategy Mycobacterium tuberculosis adopt in dormancy, how this Megadalton protein synthesis machinery assembles inside the cell, and protein synthesis in mitochondria and apicoplast of Plasmodium falciparum that causes malaria. With this, I thank you for your kind attention. Thank you, and back to you, Dr. Bhaskar.
Thank you, Dr. Kaushal. That was a wonderful overview of structural biology. I'm sure the students who are interested in this area will definitely get in touch with the respective scientists. Next, to acquaint you with the area of infectious disease biology, an area that attracts most of the attention that we have today in science, we have with us Dr. Anil Thakur, who will, in a very simple language, tell you about infection and disease, and then what RCB is doing in that area. Over to Dr. Anil Thakur, please.
Thank you, Dr. Deepika Bhaskar. So, good morning, all of you. Welcome you all in Vigyaan Yatra. So, I'm representing the infectious disease biology program of RCB. So, in RCB, we have two main missions. So, we are dedicated to advancing the path-breaking research and also committed to provide world-class education. For this mission, we have dedicated scientist teams. Those are working on different diseases and understanding their mode of actions for disease development and also identifying the drug target to eradicate these diseases from the world. So what are infectious diseases? So we can simply say a disease caused by an infectious agents like virus, bacteria, parasites, and fungi. So this is a global problem. If infectious disease arise in one part of the world, it can spread all over the world, like recent pandemic of COVID-19. Even in the past, infectious disease biology were the leading cause of the death, like large epidemic of smallpox, TB, syphilis, cholera, plague. They are responsible for millions of deaths. The influenza pandemic in 1918 alone is responsible for the 40 million deaths. Thankfully, discovery of antibiotics, immunization, and food safety, they reduce the infection. Even now, the infectious diseases are 3 top 10 causes of the death around the world or in India also. So the diseases like HIV, AIDS by viruses, diarrheal diseases like bacteria salmonella, malaria by parasites, and TB by mycobacterium tuberculosis affects millions of the people around the world. And the new diseases like Ebola, MERS, and COVID-19, they are emerging all over the world. And the situation again became complicated after the emergence of escape pathogens. They are multi-drug resistant pathogens, and none of the available drugs are working against that. And similar situations with the antifungal resistant strain of Candida and Aspergillus. And these are responsible for hospital acquired infections and nosocomial infections. And these are very alarming situations, and we need a new therapeutic targets and new drugs to eradicate these pathogens. So now, how do people get infectious diseases? For this, mainly three components are interact with each other to spread the infectious diseases. So we can simply say that the epidemiological triad, the first agent is the infectious agent, which is responsible for disease, and second is host, and third is environment. And all these three components interact with each other for the spread of the diseases, like direct way by touching, coughing, and intercourse, and the indirect way like intermediate, like touching of contaminated objects like toys, water, food, and vectors like mosquitoes. Now let's see what happened with the infections. When infectious agent infect any humans, after sometimes they get the symptom, those can be mild or severe. So when organism enter the body, some people get it off by their immune systems, but persons with weak immune system, drugs, and elderly with diseases and with no immunity, they develop the symptoms. And the time from the infection to the development of symptom is known as incubation periods. So infection diseases are multiple types, like zoonotic diseases. These are the diseases of animal, but they can spread to the humans, and around 60% of infectious diseases are zoonotic diseases like leptospirosis, toxoplasmosis, Campylobacter, and rabies. So another diseases are like emerging infectious diseases are new. These are the first time appeared in the populations, and some diseases are already there, but they are rapidly increasing now, like MERS-CoV, Ebola, Hendra, and COVID-19. And the neglected tropical diseases, they mainly affect the poorest country in the world and some of the developing countries, although they are not gaining much attention, but they are also responsible for illness like leishmaniasis, trachoma, and sleeping sickness, and schistosomiasis. The another diseases are vector-borne diseases. So these diseases spread from the infected human or animal with the help of vector to the another humans. The vectors are like ticks, flies, mosquitoes, fleas, and snail. And examples are dengue, malaria, and sleeping sickness. Around 70% infectious diseases are vector-borne diseases, and it affects responsible for the million of deaths. Now, question how we can control. To control the infectious disease responsibility of all of us. First, we can say like behavior. By changing the behavior to reduce the exposure to the infectious agents and prevent their spread, and the surveillance. Monitor the disease and detect the outbreak. The vaccines. Vaccines are the biggest success of public health, reduction and eradication of the diseases. And regular childhood immunization saved countless lives, and medications like antibacterial and antifungal kills and stop dividing the pathogens. And in others, we can say like strong healthcare systems and educations. And infection control is a develop and evaluate the disease control program, and we have to clean our environment safe and maintain the hygienic situation. So these are all of our responsibility to control the spread of the diseases. And the final weapon against these diseases are vaccines and medications. This is the responsibility of researchers to develop them. So now, but we are in surviving the valley of death. We can say the researchers are working at the benches and And the clinicians and physicians, they are taking care of the patients, but there is a large gap in between the research at the bench to the bedside, and people are dying in this valley. So we need to reduce this gap from the bench to the bedside to get out of the valley of death. So in RCB, we have a strong infectious disease biology group. So I will explain their research interest very briefly one by one. So Professor Sudhanshu Bharati's lab, they are understanding the replications of the Japanese encephalitis and chikungunya viruses, and also understanding the receptor binding and entry mechanisms. They are identifying and validating the drug target against viruses, and also developing the therapeutics and improve the vaccine efficiency that can reduce the mortality and morbidity rates. So another lab of Dr. Manjula Kalia, and she is working on the JB viruses, and this virus affects a lot of children in India, and several of them are died, but those survive, they can get lifelong neurological disorders. And in her lab, they are identifying how this JB interact with the host cellular immune system and the disease severity. That will help her to identify and develop for the development of therapies and vaccine and antiviral drugs. So another group of the Dr. Aru Banerjee, they are understanding the immunopathogenesis of dengue virus infection. So dengue virus cases in India are increasing every year. And the dengue virus infection is responsible for severe plasma leakage, severe hemorrhage, and severe organ impairment. And in his lab, they are understanding the interaction of viruses with the immune system and their impact on disease development. And they are also identifying the prognostic marker of virus infections and disease progressions for therapeutic purpose. The lab of Dr. C.B. Srikant, they are understanding the molecular mechanism governing the gastrointestinal infections with Salmonella. Though Salmonella is a prototype of model organisms, to understand the range of diseases, and his lab, they are dissecting signaling pathways such as post-translational modification, epigenetic program, and immune signaling during the host pathogen interactions. So the lab of Dr. Prasad Abhinavi, they are using the Planarian model system to understand the adult stem cell regeneration during pathogenesis and after pathogenesis. And this knowledge will help him to design strategy to control the adult stem cells regenerations during infection to promote the regeneration of the infected damaged tissue. So in my lab of Laboratory of Protein Translation and Fungal Pathogenesis, I'm probing the protein translation process of pathogenic fungus like Candida albicans, Candida glabrata, and Candida auris to identify the novel and therapeutic targets for the treatment of fungal infections. So I'm ending here my presentations with this quote: "Nothing in life is to be feared. It is only to be understood. Now is the time to understand more so that we may fear less." So ideologist has predicted in future we have to face more pandemics like COVID-19. So now is the time to get ready to face them. For the students, join the team at RCB and face the challenges together and make this world happily place to live. Thank you all.
Thank you, Dr. Thakur, for a very inspiring talk on the area of disease biology and the diversity of work that's going on in RCB in the area talks of itself and what may motivate the young to join us in this area. Next, we have with us Dr. Gitanjali Chawla, who will tell us about another very important domain of biotechnology, namely molecular medicine, and what are the sub-areas in which the scientists at RCB are working in this. So over to Dr. Gitanjali Chawla, please.
Thank you. Can you hear?
Yes, we can see your slides and we can hear you. You are on mute, Dr. Chawla.
It's almost noon, and I hope all of you are having had an interesting session so far. Today, I will be talking to you about the research programs in the molecular medicine theme at Regional Center for Biotechnology. But before I get into the specifics of the research programs in the next 10 minutes or so, I'd like to give a very brief introduction to what is the study of molecular medicine. So it is the study of cellular and molecular phenomena that in biological systems, and enhances our knowledge about human diseases, and ultimately aids in the design for or discovery of tools for either diagnosis, for prevention, or for treatment of diseases. And its birth dates back to 1949, when Linus Pauling, who was a very great scientist, and his colleagues published a paper in the Science magazine that actually showed for the very first time that a molecular change in our DNA could lead to a defective protein, and that protein was hemoglobin. And this defect in a protein was actually responsible for a disease known as sickle cell anemia. So coming to RCB, there are 5 independent or principal investigators under the molecular medicine theme, and they lead 5 labs which have different diverse scientific interests, and they use different experimental approaches and different animal model systems to try to probe at a molecular level different normal processes and the diseases associated with them. So we have these are the list of, or the names of, all the 5 labs. We have the disease biology lab, the lab of functional proteomics, lab of developmental genetics, lab of neurobiology and genetics, and the RNA biology lab. So now I will tell you a little bit of what goes on in each of these labs. So the disease biology lab is led by Professor Prasenjit Gujet, and his lab studies systemic inflammation in human diseases. And what is inflammation? It is our body's, is a normal response of our body's immune system to any infection or injury or a toxic material. And immune cells rapidly respond to the area or damaged area and actually try to fix the problem. And in doing this, there is increased blood circulation, there is release of proteins and antibodies, and that leads to pain, warmth, swelling, or redness. Usually, this response lasts for a couple hours or a few days in case of acute inflammation. However, if it is left untreated, it actually keeps the body in alert for a long period of time and is then referred to as chronic inflammation. And this can lead to damaging of normal cells, tissues, and organs. So if left untreated, this chronic inflammation can lead to development of diseases like arthritis or cancer or even type 2 diabetes. So basically, Professor Prasenjit's lab is trying to understand what are the molecular mechanisms underlying chronic inflammation in different disease contexts. So they're looking at acute mountain sickness, type 2 diabetes, accidental trauma, viral infections, and autoimmune disorders. And one approach that the lab uses is to look at interactions between different immune cells. The second lab in the molecular medicine theme is the lab of functional proteomics. It is led by Dr. Tushar K. Mehti, and his lab studies protein quality control systems in neurodegenerative diseases. So, and what are neurodegenerative diseases? So these are diseases in which there is a progressive loss of structure and function of cells known as neurons in different regions of the brain. And one of the most prevalent neurodegenerative diseases is Parkinson's disease, in which patients have a difficulty in walking, they have tremors, there is loss of balance and coordination, and also difficulty in talking. So one of the key hallmarks of this disease is the accumulation of improperly folded or misfolded proteins, both outside and within cells or the neurons which are present in a particular region of the brain. And the region of the brain that's affected is brain stem. And this is the region that allows us to move smoothly and also stabilizes our balance. So normally we have different machineries in our bodies that can detect these improperly folded proteins and eliminate them from the system. But these are basically autophagy or lysosomal degradation pathways, or they are the ubiquitin proteasomal pathways. So Dr. Tushar's lab is interested in trying to understand in what happens or how these proteins get misfolded, and if there is and what are the breach that happens in these different normal machineries that lead to disease. And one particular class of enzymes that they're interested looking at are known as deubiquitinating enzymes. The third lab in the molecular medicine theme is Lab of Developmental Genetics. It is led by Dr. Sam J. Matthew, and his lab studies control of muscle development, stem cell regeneration, and disease. So skeletal muscle is extremely important for locomotion, for posture, for support, and for metabolism. And Dr. Sam's group is interested in finding out what are the signals that regulate muscle differentiation. So by understanding what are the signals required for proper differentiation of skeletal muscle, his group is trying to understand how improper differentiation leads to either congenital diseases or cancer or regeneration after muscle injury. The fourth group in the molecular medicine team is the Lab of Neurobiology and Genetics, and it is led by Dr. Pinky Kane Sharma. And her group is interested in understanding taste and its modulation using Drosophila. So taste is the ability to recognize different flavors in food, and it is extremely important because it helps us to evaluate the quality of food. For instance, high calorie foods are sweet, and it also helps us to avoid toxic substances, and in some cases, bitter substances are toxic. So now it is well established and published in several books that taste sensitivities map to specific regions of our tongue. So we have the sweet and the salty, which is coded by the front part, and then you have bitter at the back part and sour at the sides of it. So the key question is, how is taste? How do we perceive different tastes? So taste signals begin when food particles interact with certain proteins known as receptor proteins on taste buds. And once a food particle is recognized by the receptor, it orders the taste buds to send a small electric current to the brain. And nerves then carry these impulses to different regions of the brain that allow us to perceive different tastes. So Dr. Pinky's group is using an invertebrate simple model system, Drosophila. Why? Because it can taste organic molecules in the similar range as humans. So they're trying to understand what are the brain circuits that regulate feeding behavior, and also how taste is modulated by different factors such as thirst or starvation or in different diseases. And the goal of the lab is to, by identifying these genes or pathways or neurons that are required or that regulate feeding behavior, Dr. Pinky's lab is trying to develop strategies for treatment of eating disorders. The fifth group in the molecular medicine theme is the RNA biology lab. It is led by me, and we are trying to understand the role of diet in aging processes. So aging is the process of getting old. It leads to cellular and organismal dysfunction and an increased risk for a number of diseases. So as we grow older, we are also more prone to a number of diseases, such as the cardiac disease or diabetes, cancer, dementia, Alzheimer's disease. So my lab is trying to understand what are the factors that regulate the rate of aging so that we can modify it and not only enhance lifespan, but also push these diseases to a much later part of life. And one particular intervention that has been shown to influence the rate of how old we get or how healthy we are as we age is diet. And we are trying to understand what are the genes regulated by diet so that we can modify these genes, or by modifying different or using different diets, we see what genes are modulated, and the genes that we work with are do not code for proteins, but for RNAs known as microRNAs. So our hope is that if we can identify what are the genes or pathways that are regulated by diet, then we can not only find a way or a pill or a therapy, not only to increase lifespan, but to also treat all these diseases that come in a package all at once. So with this, I come to the end of the molecular medicine theme. And I'd like to say that this is a very broad field, and it utilizes experimental techniques from physics, from chemistry, from biology, from medicine, from bioinformatics, to aid or to describe molecular structures, to pinpoint molecular and genetic errors in disease, and to try and design strategies to fix these errors. So, and I thank you for being a part of this program. And now I'll hand it over to Dr. Deepika Bhaskar.
Thank you, Dr. Chavda, for walking us through this extremely diverse area of molecular medicine and different aspects of research that are being done in RCB. RCB also has a very vibrant group in the area of cancer biology and therapeutics, where our scientists are trying to study the molecular mechanism underlying the cancer development and its progression, foraying into cancer therapeutics, knowing about biomarker, diagnosis, the therapeutic targets, drug discovery, and studying the delivery vehicles. To tell us more about this area, we have with us Dr. Rajendra Motiani, who will tell us more about this area as well as about cancer biology being studied at RCB. Dr. Motiani, please.
Sure. Thank you, Dr. Bhaskar. Good afternoon, everyone. And on behalf of RCB Fraternity, I would like to take you through our cancer biology and therapeutic program, which is an exciting program. But first of all, I would briefly like to take you through the key facts about the cancer and why do we actually care about cancer. Um, so if we look at number of deaths by cause, cancer is the second largest deaths causing factor. Close to 1 crore deaths annually are caused by cancer. Therefore, it's a key role for scientists to understand what drives the cancers. So this is the world number, and the story from India is not very different. And actually, if we look at the very recent data from 2020, 1 in 9 is actually at risk to have a cancer during the course of life, which is a very significant number. And considering India's population, we are looking at alarming numbers in coming future. And one of the reason which is actually connected to the cancer is increased longevity. And the facts that are coming up are, with the increase in the age, the chances of cancer associated deaths are increasing. And why is it so? Why cancer is associated with aging? And if we look at the root cause, what actually happens that most of the cancers are associated with genetic changes. And these changes are acquired during the lifetime. So when a child is, growing older, by the time it reaches to old age, then there are number of changes that have happened, and these genetic changes predisposes to the cancers in life. And what are the other causes? Heredity, radiation, chemicals, occupational viruses, tobacco consumption, and uncontrolled cell divide. And another factor that is playing a key role in cancer progression is immune evasion. So typically, immune cells can attack a cancer cell and thereby control the cancer growth. But many a times, these cancer cells are able to evade immune cells, and this evasion leads to cancer deaths and metastasis. So what is metastasis? So most of the cancer associated deaths are associated with metastasis to the secondary sites like liver and lung. And this spread is completed by primary cancer going through either blood or lymph nodes, reaching to the secondary metastatic site and forming a new tumor at this site. And most of these deaths are associated with these secondary tumors because they are difficult to control. And why it is difficult to treat cancer? Because one single pill or a treatment strategy doesn't work for all. And this is because there are different DNA changes or genetic changes that can predispose to same sort of cancer. And that brings us to the era of personalized medicine, wherein if we look at the problem, typically same sort of therapy is given to a group of patients, but not all of them respond to the treatment in the same way. Some of them respond very favorably, some of them don't respond, and few others can show some adverse effect. So the future is to get into more personalized diagnosis and treatment, and for that, lot of basic research has to be carried out to identify why these individuals, although they are demonstrating same sort of cancer, the underlying cause is different, and one has to identify these causes so that individual therapy or personalized therapy can be given to these patients, so that overall we see a positive effect in all the patient population. And to understand that, one has to really understand what are the cellular and molecular signatures that drive cancers. It can be ranging from increased proliferation, evading growth suppressors and immune systems, metastasis that we talked about, continuous replication, and angiogenesis, because blood supply is required for nutrition and oxygen. So increased angiogenesis also contributes to this cancer progression. So we at RCB are trying to understand both these cellular and molecular processes, as well as some groups are trying to develop therapeutics to take care of cancer progression. So I will take you through the different group activities. So at Laboratory of Calciomics and Laboratory of Cellular Dynamics, led by Dr. Shivram, They are interested in understanding the process of cell division and intercellular communication. And what they are actually trying to understand that, during this cell division, how do these chromosomes they segregate from the metaplate, and other area of their interest is how do cells crosstalk with each other through small tunnels which are nanotubules, and through which the communication is driven through growth factors, mitochondria, and other things. So their idea is that if they can understand the basic biology of these processes and target them, that could help in future for treating cancers and other pathogenic transmissions. The second laboratories works around biology of centrosome and cilia, wherein again centrosomes they play a very important role at the time of cell division, and it is important that these centrosomes and cilia function in a very regulated way, so that a proper chromosomal disaggregation happens. And in cases of misaggregation, what happens? Aneuploidy happens, and as we know that it is one of the important factor leading to cancer. So the group led by Dr. Karthikeyan is interested in understanding the defects in the structure and functions of centrosome and cilia, and to eventually use the strategies to correct these errors for targeting cancer progression. Next, we have a laboratory of calciomics and systemic pathophysiology, wherein we are trying to understand what drives cancer metastasis. And if we look at this figure over here, when normal cells are transformed to cancer cells, one hallmark signature is upregulation of calcium pumps, channels, and transporters, both at the plasma membrane and organelles like ER and mitochondria. This leads to elevated or different signatures of calcium, which are taken by the nucleus and the other effector proteins to drive carcinogenesis. And what we are trying to do, we are trying to design drugs or identify drugs that can be used to tackle either the expression of these channels or these calcium signatures. The other area of our interest is how do these cells metastasize? As we discussed, the primary tumor is not the main cause of death. Most of the cancer associated deaths are due to metastasis. And an important phenomena that happens that, cancer cells, they enter blood vessel and they come out of blood vessel at the secondary site. What we are trying to understand how these cancer cells enter into the bloodstream and how they are able to come out, understanding the metastatic process. And we are looking at mitochondrial biology in this area. The long-term goal is to develop therapeutics for targeting cancer metastasis. Finally, we have group of nanotechnology and chemical biology led by Dr. Avinash Bajaj, wherein his group is developing strategies for cancer drug delivery as well as combination therapy and getting into future of precision cancer medicine. So over here, some studies that I am sharing is, what their group was able to do. They were encapsulate 3 different drugs targeting angiogenesis, proliferation, and migration in one single gel. And because of that, the tumor volume in mice was significantly reduced, as well as the survival of these mice was enhanced almost twofold. And they are also interested in understanding how these drugs, wherein there are multiple drugs are encapsulated in a single gel, how they work at the molecular level. And long-term goal is to develop technologies for combating cancer and to develop strategies for site-specific localized chemotherapy. Uh, for doing cancers, we need high-end animal facilities, and because we are interested in understanding the how this cancer is spread in organs, we use zebrafish and mice, and RCB hosts a very high-end zebrafish and a small animal facility, mouse facility, where we are able to actually look into how these cells metastasize and cancer spread is achieved. Um, with this, I would like to say that cancer research saves lives, and I encourage young minds to join RCB in our exciting cancer biology program. And with this, I would like to thank you all for your attention.
Thank you, Dr. Motiani. That was indeed a very vibrant picture about what cancer research in RCB is all about. And I'm sure the students who want to work in this area will definitely contact the scientists for the same. You all must be wondering that there is one domain of biology that we have not talked about, and that is plants. working on plants and the research on plants in conjunction with what enhances the agricultural practices is a very important area to which also RCB contributes for addressing global challenges like food security, environmental protection, and finding alternate fuel sources. To know more about what RCB does in the area, I am handing over the platform to Dr. Ramu Vemanna, who will get the young excited towards this very important domain of science and biotechnology. Dr. Ramu Vemanna, please.
Thank you, Dr. Bhaskar. So I am here to represent Agriculture Biotechnology Unit in RCB. Presently, we are worrying about food availability, hunger, and malnutrition. If you go with a packet of money, you will feel that large amount of groceries or the vegetables are available in the market. So, but we have to ask a question: that is this food is sufficient to feed the world, or those who are not affordable, or is this food is really nutritionally rich, or this food is really free from diseases or contaminations? If you see the global hunger index, India is about 102nd position below Pakistan, and this is really worrisome. And India is also facing a problem of malnutrition, especially child malnutrition and also pregnant women malnutrition. So because of lack of availability of food, the rural India is eating less than it did in 40 years ago. And another major concerning factor is affordability is leading to wastage of food. Apart from these challenges, agriculture scientists are facing the uneven rainfalls leading to the drought, insect infestations, fungal and bacterial infestations that affects crop growth and development as well as productivity. If you see, our colleague talked about green revolution. The green revolution really made an impact to improve the food productivity. However, up to 2000 years, the green revolution surpassed the population was existing. After that, population is keep on growing. However, the growth of the productivity of wheat and rice did not grow. So now we need a golden revolution to improve the yield as well as the quality traits. So in these conditions, how do we increase the yield of the crops? There are two approaches. One is the traditional conventional breeding approaches, where we identify a resistant genotype and identify yielding crop varieties and cross both of them and identify the near isogenic lines. And several years after that, you will identify a different crop or a new yielding variety or hybrid variety. That takes several years. However, there are new technologies, that is molecular genetic approaches. By identifying a new one gene is responsible for virus, insect, or pathogen, and you can transfer that one gene into a different genotype, that is a yielding genotype. Within a short period of span, you can develop a new variety. So how we can develop these new varieties are, so you have to identify a trait of your interest. If you want more carotenoids or more nutrition, you have to identify the genes which are responsible for the trait from plant or from microbe, and isolate DNA and prepare the mRNAs which are responsible for it, and subsequently develop a few specific vectors with a suitable promoters. And these promoters can be incorporated into agrobacterium, where agrobacterium can infect the plants, and develop callus. Through tissue culture approaches, you would transform these gene constructs into the plants and identify a real transformants, and those transformants will give you superior phenotype. This is the general processes which is involved in developing superior plants. So some of the examples like these how biotechnological interventions are giving a societal impact is BT cotton is a good example. BT cotton has been developed using cryogens, which is acting against lepidopteran insects. After the incorporation of Bolgard 1 and Bolgard 2, the India's cotton production rates was drastically improved. Presently, India is the largest producer, and now we are exporting the cotton at higher rates. So another recent example is BT brinjal. All of you are aware of that. So BT brinjal, now Bangladesh is adopted to grow BT brinjal, and they're getting more benefit, and more farmers are getting usefulness of this technology. And herbicide tolerant corns has been developed using EPSPS gene that is acting against the glyphosate or Roundup herbicides. And because of this intervention, the farm women are getting benefited because their health is improved drastically. So to reduce the malnutrition, golden rice has been developed, that is vitamin A rich rice, especially targeting phytoin synthase and phytoin desaturase genes, which are involved in carotenoid biosynthesis. So the recent example of biotechnological intervention is adopting CRISPR-Cas9 technology. So generally mushrooms or the apples, when you cut them, they develop a browning phenotype because of there is an enzyme called polyphenol oxidase that is responsible for browning in both mushroom and apple. By using CRISPR-Cas9 technology, polyphenol oxidase was edited, and then the mushroom is no more turns to brown. So these mushrooms are now deregulated for the consumption in USA. So these are the technologies where we can adopt for the betterment of the societal impact. We at RCB do basic and strategic research. We try to address bacterial pathogens, fungal pathogens, and abiotic stresses like drought. And one group works on plant cell wall aspects to convert the biofuel. We use Arabidopsis, Nicotiana benthamiana, Medicago truncatula as a model plants, and pea, rice, and poplar as a target plants for the improving the biofuel and other aspects. Dr. Seket Bhattacharya at RCB is trying to understand signaling molecules which is responsible for the plant innate immunity mechanisms. When bacterial pathogens or bacteria infect the plant system, they secrete particular proteins called effectors. These effectors trigger central regulatory assembly molecules and also trigger effector mediated perturbations. And that leads to, there are a lot of changes in the signaling events. So based on these events, is trying to understand signal transductions in innate immunity and crosstalks on growth and developmental networks. And also is trying to elucidate the signaling routes that how field tested molecules are for giving a resistance towards the crop protection. Is also trying to understand how pathogen effectors functions in planta and what strategies to counterattack those pathogen effectors. This group is also interested to identify post-translational modifications to immune modulating and perturbations. So our colleague Dr. Divya Chandran is trying to understand investigations into the molecular mechanisms underlying legume powdery mildew interactions. Powdery mildew is one of the biggest problem in many legume crops because they completely damages the crops. She works on obligate biotrophic pathogen RCP that causes powdery mildew. She's trying to understand how this pathogen is causing the disease in pea and medicago. She's trying to understand the each and every step of the pathogen infection and penetration to the plant. And she's trying to identify the host and resistance susceptible factors. Especially she's interested to identify secondary metabolites that provide resistance and sugar-related mechanisms, host nuclear positioning mechanisms, and plasma membrane sporulation mechanisms. If she can understand these things, she can develop better strategies to come up to protect the plants from fungal infections. She's also interested in identifying the RCPPC effectors that cause disease. And finally, she's trying to develop a new durable resistant crops by using biotechnological approaches. We at RCB, our group, we are trying to understand about drought stress adaptation mechanisms. When there is a shortage of water, plants experience a stress, and that leads to wilting of the plants. Our cell turgor is reduced. So under less water potential, ROS or carbonyl stresses get increases more, that affects protein synthesis and all. So our goal is sustaining protein turnover because proteins are essential for the food. And to improve protein sustainability, we target enhancing the detoxification enzymes, deciphering the roles of ribosome biogenesis factors. Especially we are interested in extra ribosomal functions of ribosomal factors and identifying small molecules that trigger plant stress tolerance as well as crop protection to improve the plant health. And we are also investing our time on creating genetic variability in rice because genetic variability is essential for the many agronomic traits. Our global agenda is more crop per drop. Our colleague Dr. Prashant Pawar is working on improving plant cell wall to convert into the bioenergy applications. Generally, plant cell wall is made up of very complex structures and is trying to develop new altered plants with simple cell wall structures. Because of simple cell wall structures, more ethanol and value-added products can be developed. This group is interested to identify plant cell wall biosynthesis and assembly factors. Is trying to characterize the role of GDSL lipases, esterases families, and also trying to characterize polysaccharide esterases and transferases. So by identifying these things, you can identify the interactions between plant cell wall components of these things. His group is also interested in converting recalcitrant lignocellular biomass by using simultaneous reduction of xylon and lignin monomers and modifying Zelan side chains to reduce cross-linking effects, and also spatiotemporal alterations of plant cell wall modifying enzymes. He's also trying to understand why dwarfism is happening in the plant cell wall mutants and identifying the sensors and their role in maintaining cell wall integrity. To do all this research, we at RCB has all the facility required for plant research. We have plant growth, walking growth chambers, and newly constructed climate controlled greenhouse facility. So we provide short-term, long-term trainings, PhDs, postdoc programs are available. I encourage all the young minds to join our groups to solve global hunger index and nutritional problems. I would like to end my talk with your body is a temple and you are what you eat. And with this, over to Dr. Deepika Bhaskar. Thank you.
Thank you, Dr. Ramavemanna. I'm sure with such a encouraging talk, many students will be motivated to work in the area of plant biology, and looking at the excellent facility that RCB offers will definitely consider RCB as their future destination. If we want to study the complex networks of biologically relevant entities, we need to do a systems approach to biology. And if we want to rewire and reprogram organisms for our benefit, the approach is synthetic biology approach. To tell us more about this area, we have with us Dr. Kinshuk Raj Srivastav, who will walk us through the area as well as tell us about the research that is being carried out at RCB in the area. Over to Dr. Kinshuk, please.
Thank you very much, ma'am. And good afternoon, everyone. Today, I'll be representing the synthetic biology research group that is happening at the Regional Center for Biotechnology. And in the due course of my presentation, I'm going to discuss with you what synthetic biology is, what is its future prospect, and what we do at Regional Center for Biotechnology. So synthetic biology. This is a very young discipline and just established 10, 12 years ago in the world. And there is no official definition of the synthetic biology is yet. So before I define, let me first briefly introduce you or give you an overview of the traditional discipline that gave a birth to this new discipline, the biology and the engineering. Biology studies life, specifically the organism and their relationship to the environment. And in engineering, engineers solve the problem using the science and math, following the engineering principle, design, build, and learn to solve the problem. And the fusion of these two gave a birth to a new interdisciplinary science, which is called the synthetic biology, that applies the principles of engineering, that is design, build, and learn, and uses the available information about the microorganism or any living organism, and uses the developed the genetic manipulation tool, molecular biology tool, to engineer the organism to evolve a new organism to carry out new function. So an analogy is that engineer builds a machine with using the electronic circuit. Synthetic biologist engineer the nanobio machine with the simple biological circuit. And the component of this simple biological circuit, that is the pieces of DNA that may come from a very diverse origin of life. And then these pieces can be stitched together to form a very simple network that can be perfectly integrated into the already existing complex network of a native host to evolve a new function. For example, here the microbial host is now engineered by grafting the new circuit to accept the lignocellulose biomass and convert it to the biofuel. So the two principal approaches exist, the top-down and the bottom approach of the synthetic biology. The top-down approach, we do a minimal stripping or fabrication or replacement of addition of a new entity into the native host to generate a new machinery for performing a new function. In the bottom-up approach is a very new, that is, de novo approach, where you have the Lego, such as the building blocks, bricks, such as DNA, protein, and lipid, and you assemble a minimal artificial cell to carry out a new function. So it appears that this synthetic biology is very, very fascinating, but the question remains, what significant advantage that this new discipline can bring to the community for its welfare? So the most strikingly I can think of these programmed nanobiomachinery cells. They can amplify a lot and make a copies of itself in a very less span of time. So therefore, these nano bio factories can make a lot of product in a less span of time and can fulfill the supply and demand problem. Second, this biological machineries already there are complex network exist and carry out a complex task. We just have to tweak a little bit to trigger a new function, and we can start using this programmed biological machinery for the new function. The third is, since in the synthetic biology we are using the microbe and any other living organism to produce the compound. So therefore, since we are using this vehicle, the compound produced will be non-toxic, otherwise cell will not survive. So therefore, synthetic biology provide an eco-friendly solution to many difficult problems. And the most important thing is this new, although this is a very new discipline, it has started contributing a lot to the global bioeconomy. As you can see here, in 2018, 5 billion to the projected in 2022 is a 40 billion contribution to the bioeconomy overall the world. And there are a lot many top players in ranging from the different sector: healthcare, food, software, agriculture, biofuel. And in addition to the top players who are contributing to the economy, there are many startups are getting established, and they are attracting a lot of billion dollars investment through their startups. So with this, we now know that there is a huge emerging applications of the synthetic biology. Here I can just mention a very few. For example, in case of energy sector, a custom built microbes are already generating the hydrogens and the fuel and performing the artificial photosynthesis. In medicine sector, a lot of drugs, vaccine, and diagnostic platforms are getting developed by implementation of the synthetic biology platform. In environment, detection of the pollutant and their bioremediation is happening at a rapid, promising and going to happen at a rapid pace. In chemical industry, a lot of fine chemical synthetic fibers are getting produced through the synthetic biology platform, and in agriculture and novel food additives. So now we know that this synthetic biology is a very emerging and is taking a very good shape in terms of revolutionizing the way we do science and engineering. So now, as I mentioned, that now synthetic biology is a very new discipline in the world. Our entity, that synthetic biology and system biology division at RCB, is also very young. It's just an year old, and we are the three key players coming from three different backgrounds and joining hands together and trying to solve this real problem. I myself, Kinshuk Raj Srivastava, working in the area of the biocatalysis and enzyme engineering, Dr. Nidhi Adlka working in the system and synthetic biology, and Dr. Ambadas Rode works in the RNA synthetic biology. So I'll begin providing you information about what I do, and then I will walk you through what other faculty colleagues do at RCB. So my work is focused on development of microbial platform for drug discovery and development. And I use the engineering principle, which I mentioned before: design, build, test, and learn, for providing a greener process and a better product. And my research begins with a lot of database mining using the chemo and bioinformatics software, chemical databases, drug databases, protein databases, and genome database. And all this mining, information mining, provide us a clue or give us an insight, better insight for designing a smart library of a drug and to construct the minimal biosynthetic or biocatalytic pathway that will be used for expression into the heterologous host for production of a compound. So effort to little bit elaborate further on the strategy, we exploit the plug and play synthetic biology for drug discovery and development. We use the microbial hosts such as E. coli and yeast, and then the synthetic pathway that we develop, as I mentioned in the before, we redesign that for streamlining expression, cloning and expression in the pre-engineered plug and play host. Further, we transform this into the heterologous host such as bacteria and yeast, and then we optimize the strain and the process for large-scale production of a desired compound. Another very different approach that my group is taking and evolving and designing artificial enzyme. And now what does I mean by artificial enzyme? That enzyme that never existed in nature. So in other traditional approaches, we use the genetic material or enzyme that exists in the nature, and we put it into the pipeline and try to evolve a new function in the microorganism. But in this particular case, we are designing a totally new enzyme to provide an innovative biocatalytic solution for traditional industrial scale production process. So what we do here is the chemical processes are very hazardous. So what we do, we use the chemical catalyst, and then we graft into the protein scaffold, evolve a new enzyme to carry out new function, and then we integrated this enzyme into with other pathways already present in the microbe to evolve a total new reactivity and the new transformer that nature never visited before. So now over to Dr. Nidhi Adalkas. Her work is focused on the system and synthetic biology for the bioproduction. So let me briefly introduce what does the synthetic and system biology mean. These two are the sister discipline. The focus in the system biology is to understand better using the computational and experimental tool how the genetic networks and the proteomic networks work in the microorganism or any living organism. And what the synthetic biologists do is they uses the available information to engineer the native host to produce a new biological system to carry out a new function. So Dr. Nidhi Adalkas' work start with the genome and proteome based modeling and analysis, followed by the pathway engineering and the process engineering, further biofermentation and development of bioreactor and scale-up technology for the production of a desired compound. Dr. Amba Das Rode, his work is focused on the rational engineering of a biological system, and he uses the riboswitches as a tool for ligand-dependent temporal and spatial control of a gene expression. What is a riboswitch? Riboswitch is the RNA regulatory element that binds specific ligand and control the gene expression by the conformational switch, and that activates the translation of the reporter protein. And his work has a lot of application in the diverse fields such as biofuel, pollutant degradation, and therapeutics. So before I end my talk here, I want to bring an important point related to the career is the synthetic biology is a very exciting career for the golden future. And why it is so? Because I briefly touched this, that the synthetic biology field is a very nascent, but this market is projected to reach 20 billions by 2025, compared to 6.8 billion in 2020 this year at a 23.9 CAGR rate. And in addition, this contribution to the economy is coming from the big industrial player. And in addition to the big industrial player, a lot of startup is getting established in diverse sectors such as healthcare, industrial, chemical, biofuel, organism and engineering and automation platform, and consumer product, etc. So now the question is, what skill do you need to get into this territory? Certainly, since this is a multidisciplinary domain, so the synthetic biologists should be grounded at least in one or the several core disciplines, naming genetics, system biology, microbiology, and the chemistry. And they should have the engineering mind set up to start building a new organism to do a new function. So now I end my discussion and over to Dr. Deepika for further discussion.
Thank you, Dr. Kinshuk. That was a very good representation of the area, and I'm sure many students would be excited to work in this area in future. RCB not only has state-of-the-art research laboratories, it also has a very high-end facility with the most sophisticated equipment to take care of the most advanced needs of the scientist or anyone in India who wants to use this facility to answer some interesting research questions that anyone may have. We call it the Advanced Technology Platform Center. To tell us about which, we have with us Dr. Nirpendra Singh. Dr. Nirpendra, please tell the audience about this facility and how everyone can use it. Dr. Kinshuk, please stop sharing your screen. Over to Dr. Nirpendra, please.
Just is it visible now?
Yes, it is visible. Please
Okay.
switch on the slideshow.
Okay.
Yeah.
Yeah. So sorry for the things. I'm Nripen, and we in RCB we have Advanced Technology Platform Center, which is a 20,000 square feet space dedicated specifically for the advanced technologies which are needed for supporting the research infrastructure for whole of the RCB and NCR Biotech Science Cluster. So the major mission of this Advanced Technology Platform Center is to act as a catalyst for multidisciplinary basic and translational research by providing relevant state-of-art instrumentation and professional services for research laboratories at the NCR Biotech Science Cluster, industry and academia. And another mission of this ATPC is to train personnel in the use of these high-end technologies and to develop new technologies in collaboration with academia and industries. So at ATPC, we have these 6 technologies platform, which are basically used by all of the country. And these technology platforms are mass spectrometry, optical microscopy, protein production, genomics, flow cytometry, electron microscopy. So all these technology platforms are very much useful for most of the researchers who have talked about this in their slides, recent slides. So I will go one by one to each of the facility and tell about how this facility can be utilized in their biological functions, biological exploration. So this is the mass spectrometry facilities. In the mass spectrometry facilities, we have 3 different kind of mass spectrometer. Basically, these mass spectrometers are nothing but a device which can measure the molecular weight of any molecule which can be ionized. So if you think about any molecule, like a small molecule, maybe a water, maybe a protein, peptides, all these molecules which are charged, which can that can be measured. So these molecules have a signature because all these molecules are made up of some kind of elements. So these molecules have a markers. So it's kind of fingerprint. So when these fingerprints are subjected for a molecular weight determination, they create a signature for particular protein or particular metabolite. And we identify these particular metabolites so that those particular metabolites or protein can be identified and put into a position how they are identified. So for a simple example, it's like suppose if you have a protein or some peptide which is in a gel form, you digest it with some kind of trypsin or chymotrypsin or any kind of proteases and create a signature peptides. And these signature peptides, when they are mapped into a database, you can find out what these proteins are present, what are the protein which is there in the gel. So in that way, this mass spectrometry is helping the scientists to identify the protein, identify the metabolites, and not only that, this facility is helping the users to find out if there is any kind of post-translational modification. So post-translational modifications are the modification which is happen after the synthesis of the protein, like phosphorylation, glycosylation, simulation, and there are approximately 300 different kind of post-translational modifications. So this facility is trying to handle all those kind of modifications. This is about the protein. But if you talk about metabolites, metabolite is also a same kind of analysis where you want to know what is the M by Z of that molecule. So this facility is helping all the users for metabolite identification, and which is very much needed for basic scientist researchers. And after you identify protein, what your goal is to after identification, you want to make the protein in a very bulk quantity. So for that things, we have our protein production and molecular interaction facilities. Here we have two kind of bioreactors where you can grow your protein in a very large concentration. So bioreactor are nothing but just a machine where you grow your microorganism, where you grow your cells in a very high volume in a controlled environment where DO is controlled, CO2 is controlled, and temperature and pH is controlled, so that your cells, your bacteria can produce your protein of interest in a very high concentration. And once it produces a very high concentration of protein, what you have to do is you take out those cells and from this bioreactors and put it into a purification system, which is a FPLC system. And here, with the help of different kind of chromatography, which is affinity chromatography, maybe gel filtration chromatography, or different other kind of chromatography, you purify that protein. So once you purify that protein, what you need to do is, because as you all know, all the enzymes are protein. So all these proteins has some kind of interaction and some kind of metabolites to interact with. So you need to characterize this protein. For characterization of those protein, we have two different platforms. So these different platform, what they do is, you need to characterize, when you want to characterize those proteins, you need to immobilize those protein and find out if it is binding with those kind of receptor, is used for receptor binding or maybe antigen antibody interaction. So those kind of instruction studies you can do in these platform. And these two equipments, which is very much present in the ATPC facility, and trained regular manpower is there to help you out all those on this experiments. So all those kind of KD experiments where you want to find out whether this molecule is binding strongly with the ligand, or whether there is protein-protein interactions happening, or is protein interactome which are the other proteins are present in the interactome. So those kind of chip assays and all those you can do it in these equipments. And these are the services which is offered in this facility, where as I already mentioned, ion exchange chromatography, affinity chromatography, and size exclusion chromatography. So these are the things which can be done here in a protein production facility. So once you have your protein, then the questions come, as Dr. Prem has mentioned, seeing is believing. So we want to see whether where is your protein in the cell line system. So just for having a view on the cell line system, and I've given a representative picture of these two slides. One is a pig kidney cells imaging using structured illumination microscope. This is a super resolution microscope where you can differentiate any two molecule with a resolution of 20 nanometer. And in a confocal, if you see, this is a actin filament where the resolution is approximately 200 nanometers. So that means you can differentiate two molecules up to 200 nanometers distance. But if anything is less than 200 nanometer, you cannot differentiate these 2 points. But in a super resolution, if it is up to 20 nanometer, you can differentiate these two molecules. You can see the clarity of these two pictures and find out if these two molecules are interacting together or not. So this kind of studies can be done in optical imaging facility, which is very much equipped with all those kind of sensors and the camera. And so a lot of experiments can be done, like for fluorescence resonance scanning and fluorescence resonance energy transfer, and other kind of high content screening services are there in the optical imaging facility. This is our high content imaging system, where we have one robot and CO2 incubator attached to it. You will see this instrument in a working condition in the next video, and where this can be utilized for calcium imaging experiments, SPERAD studies, as in a cancer biology. Scientists were explaining that they were doing some kind of in vitro drug screening assay. So this equipment can be utilized there, and the virology experiments, most of the scientists are using this experiment for high throughput. When you have a whole library to screen, and you want to automate all those features, then the equipment is very, very handy. And here you can make a workflow and then submit that workflow to this equipment, and can do the work which is very much not very much possible with a single hand. So after this, because light microscopy can only give you a resolution in a nanometer, but when you really want to see something with an atomic scale, then you require an electron microscope, as Dr. Prem mentioned. So if you want to see something in a atomic scale, you need to go for electron microscopy. So if in the electron microscopy facility, we have a transmission 120 kVA transmission electron microscope, 200 kVA cryo-electron microscope, and a scanning electron microscope. So these three microscopes are used in this facility, and and you can see the representative picture. What you can see in a normal confocal microscope is a cell outline, but in electron microscope, you see in this first panel, you can see this cell line with a human monocyte cell line captured on 120 kVA. You can see the internal delicate structure of the cell, which is not possible in any of the optical microscope, and see the and now you cannot see a protein, purified protein structure in a normal optical microscope, but here in electron microscope, you can see this is a trimer structure of GP120 protein of HIV, but you can easily see the structure of that protein. And this is a scanning electron microscope of scanning electron picture of HeLa cells, and you can see the surface perturbation of all the HeLa cells. And this is the Mycobacterium smegmatis cells, bacteria, which is you can see in a great resolution with a great detailing in a scanning electron microscope. Along with this is a ribosomal image we have taken from Dr. Prem's causal lab. So this is a negative stain image of ribosome, which we took a image on normal mode in 200 kVA, and when we do a cryo-electron microscopy image of this, you can easily see this clear structure of the protein and the protein megadalton complexes. So these are the things, and after this we have a flow cytometry facilities. Now you have seen the protein into a picture form. So we wanted to see whether how these proteins and other things behave in a cell line system if you want to study those kind of things. And then we have a cell sorter system and FACS analyzer. So cell sorter system is something when you have something on your cell and you want to purify that particular cell which is expressing the particular gene of your interest or particular marker of your interest, then this cell sorter thing is very useful, where you can separate out your cells and do the studies on the cell sorters. So it could be this several lasers, and you can do a separation of those molecules. And this FACS analyzer, it also works where you cannot separate out your things, but you can do the analysis. So these two equipments are basically used for apoptosis analysis, cell surface antigen detection, intracellular antigens, monitoring of electropermeabilization of cells. If some cells have the oxidative burst or oxidative stress, then those things you can find out with this kind of instrument analysis. And monitoring electrochemical permeabilization is also very much possible in this. And after this, we have a genomic facility. Suppose because everybody is now working on a gene permutation, gene mutation, and gene cloning. So what happens is when you clone, do a PCR of your gene, you wanted to find out whether you have cloned the right person, you have you have amplified the right portion of the genome or not, or then you need to clone it and submit it for sequencing. Here we do a sequencing of that nucleotide stretch and find out if you have sequenced the right genome. And then after the sequencing, somebody wants to do a quantitation studies of some sample, some biomarker, then this is a droplet digital PCR, which can be utilized for quantitation of particular gene of interest in any kind of cell sample, whether it's a plant sample, animal sample, bacteria, or copy number analysis. So, and this is the website of ATPC, where anybody can go and book an instrument and see the charges and the contact detail. And in last 1.5 years, we have processed approximately 5,500 samples and served about almost 1,000 users. And so many institutes from Delhi NCR have utilized and all of the India have utilized our facilities. And we are regularly organizing a workshop. Until now, we have trained almost 150 people on different technologies platforms. And this is the team at ATPC, which whenever you will come, you will see and meet these people. Thank you.
Dr. Nirpen, please stop sharing your screen so I can I can play the ATPC video.
In the outskirts of Delhi on the Gurugram Faridabad highway is the huge complex of RCB Regional Center for Biotechnology. It is an institution of education, training and research with a strong focus on capacity building. It is equipped with various research infrastructures in various fields. RCB also has a dedicated Advanced Technology Platform Center, also known as ATPC. ATPC is a Department of Biotechnology, DBT, Government of India funded commercial platform that provides a medium for innovative researchers to turn their ideas into reality. It is laced with state-of-the-art research facilities, specialized personnel, and world-class infrastructure. At present, various facilities are operational in ATPC. Protein Purification and Molecular Interaction Facility. This facility aims at aiding researchers in upscaling protein production with the help of bioactors, followed by downstream processing using state-of-the-art facilities. Interactions amongst biomolecules can be studied with the help of latest available technologies like biolayer interferometry and microscale thermophoresis. This facility supports researchers and startups in their need for expression screening of the protein of interest by subcloning its gene in vectors with different tags, overproduction of recombinant proteins in 7-liter and 14-liter fermenter, protein purification by affinity chromatography and size exclusion chromatography using ActaPrime and ActaPure FPLC systems, molecular interaction studies using biolayer interferometry and microscale thermophoresis. Mass spectrometry is another important facility of ATPC. Mass spectrometry facility offers a comprehensive suite of modern instrumentations for the separation, identification and quantitation of a wide range of molecules. At present, the facility maintains 3 mass spectrometry instruments: Triple TOF 5600 Plus, Multi TOF 5800 and Q-Trap 6500 Plus. This facility provides training and services for identification and quantification of known and unknown proteins, peptides, metabolites using data-dependent DDA and data-independent DIA analysis. PTM analysis, labeled and label-free quantitative proteomics. One of the crucial facilities of ATPC is the Genomics facility. Genomics facility provides services related to microbial sequencing, pathogen analysis, biomarker detection, gene expression analysis, copy number variations, and rare mutant detection using DNA sequencer and droplet digital PCR. This facility is equipped with ABI 3500 genetic analyzer and droplet digital PCR. Another facility of great importance at ATPC is the optical imaging facility. This facility hosts 3 state-of-the-art instruments which are very rare. These are high-content imaging molecular devices, super-resolution microscope, laser scanning confocal microscope. High-content imaging molecular devices is an integrated cellular quantitative imaging and analysis system designed for rapid and automated screening of fluorescently labeled fixed and live cells in microplates and slides. The system also provides environmental control for live cell imaging, move fluids to and from cells for compound addition. Super resolution microscope and laser scanning confocal microscope. Both these instruments image fluorescently labeled live and fixed cells, animal tissues and microbial samples. The system is equipped with high quantum efficiency detectors which are helpful in acquiring images even in very low signal. Moving on, electron microscopy facility houses instruments with high performance, excellent imaging and analytical capabilities, achieving ultra-high resolution and ultra-high magnifications for life sciences samples. This facility is useful in visualizing cellular and subcellular structures, proteins, stained and unstained samples, 3D architectures of cells and tissues. All this is done with the help of volume scope scanning electron microscope and transmission electron microscope. These 2 microscopes are one of their kinds and are really amazing. Last but not the least is cryo-electron microscopy. Recent path-breaking advances have allowed the use of cryo-electron microscopy to obtain the structure of macromolecules. In cryo-electron microscopy, samples are imaged in its native condition at cryogenic temperature. Due to recent developments in detector technology and image processing algorithm, it is now possible to determine structures at atomic resolution for macromolecules. This method has revolutionized the field of structural biology and helped understand how many important biological molecules achieve function. We are sure that access to these cutting-edge facilities will help Indian researchers find new solutions to problems faced by the nation in public health, agriculture, and skill development.
Thank you, Dr. Nirpendra, for the wonderful, informative description of the facility that we provide as ATPC, one unique facility in India that can be used by any researcher across India. So please contact ATPC website or Dr. Nirpendra in case you want to use this facility and answer interesting research questions in science. RCB is uniquely positioned by having a visionary unit, the BSc BioNest Bioincubator, that fosters innovation, research, and entrepreneurial activities in biotechnology related areas. The mission of BBB is to stimulate this establishment and growth of biotechnology-based startup companies and encourage scientific entrepreneurship. To tell us more about this unit, we have with us the COO of the facility, Dr. Suman Gupta, who will tell us how you, as a with a scientific startup vision, can be a part of it. Dr. Suman Gupta, please take over, please.
Thank you, Dr. Bhaskar. So we heard about the excellent research activities that happen at RCB, and we also heard about the ATPC just before this. So now I'll be taking you through the bioincubator, which basically supports scientific enterprise coming out of either a faculty or a student or any science graduate. So bioincubators are basically the facility which handholds a startup and help them during their fledgling days and provide all relevant infrastructure which is needed to make a successful biotech startup. So the facilities that one would need in addition to an innovative idea is an excellent infrastructure, funds, and mentors during the course of the journey. So if we look at this schematic diagram, so once one has an idea with which we aim to change the existing product, service, or process, or improvise existing processes to make it more suitable or more user-friendly. So once that idea is there, one wants to test whether the hypothesis or the idea that one has, whether it has the proof of concept or not. So in order to do that, one can approach the bioincubators, which are plug-and-play facilities. So here, one gets the infrastructure, which has a lab facility, the equipments, the mentors, which are needed during the way to guide, the funds, and the networking support. So all that is provided at the bioincubator. So one does not need to spend time and energy to create one structure like that by themselves. And once you come to our incubator, you generate the proof of concept or validate your idea that you have. And if you get a positive result, you scale it up and take it to the market. And in case you don't get a positive result, then you again change something in your idea, improvise it, and again go through the process. So BioNest bioincubators are created by BIRAC. So BIRAC is one of the enterprise by Department of Biotechnology, which has created these facilities, which are called incubators all across India. So the red dots that you see here are such facilities which are available for the startups to use. So about close to 700 startups have been supported in such bioincubators, and about 200 total products and technologies have been commercialized so far. So our incubator, which is called BSc BioNest Bioincubator, is spread across a 35,000 square feet covered area. We provide lab spaces, office space, culture space, advanced equipment facility, assistance for funding, patent support. So all that, what is needed for you to generate a proof of concept for your idea is available at the incubation center, and anybody can come here and start working here. So these are few pictures of the labs that we have. We have a shared wet lab where A lot of startups share the same thing. We have an independent cubicle to be dedicated to a startup. So different kind of facilities are available, which you will also see in a video that will play shortly after the talk. So I'll now take you through some of the innovations that scientific enterprises have been able to bring the science from the bench to the bedside. So this is one such startup, which is by the name of OM. So these guys are working on patients, throat cancer patients. So what happens in a throat cancer patient that during the surgery, the voice box has to be removed. And because of this, the patient is not able to talk after the surgery. So these guys have come up with a medical device that help these patients to talk. So through this device, when they speak, they're audible. People can hear them. So this is a very good innovation and a very affordable one too. You can see that it is brought to the patients at $1 per device cost. Another example that we have is by the OrthoHeal company. So this is another startup who's working on creating a orthopedic cast, so which is unlike the traditional the cast that the fracture when somebody has a fracture. So the cast that one has, those are very heavy, they are not washable, they are very difficult to wear, and in summer that is really difficult. So these guys have come up with a cast which is based on nanogel. So as soon as one wears them on the fractured part, in the presence of sunlight, within 5 minutes, it solidifies and becomes a hard structure. And this is how one can easily wear it. So this is a very interesting innovation that is brought by these guys, and these are available in the market at various places in various states. Now I'll talk to you about 3 innovations which have come up during this pandemic time. So this is one innovation is by the company called Nasofilters. So these guys have been able to build these nasofilters, which are based on nanotechnology, which can be easily fitted in the nostrils, and this gives protection from dust, smog, bacteria, viruses, etc., and is able to filter 2.5 pm aerosols. And you can see that the cost that they've been able to provide it for is just ₹10. So this has become really affordable for everybody around. Another innovation during the COVID, for the COVID area is the surface disinfectant, which is based on silver nanoparticles. So this is effective again against bacteria, spores, and virus. It is aqueous based, so there are no hazardous chemicals which are used in this, and this can be applied to clinics, hospitals, and any surfaces that we have, the woods and the stainless steel. So this has found very great use in the current situation. One more example is by the company Sleepis. So they have been able to come up with a device which is a contactless device that measures the breathing, the heart rate, and the activity of a patient. So as you can see, there are no wires attached to the patient when this device is getting used, and the doctor is able to get the data at his laptop. So it is a contactless device which has found a very good use in the current situation. This innovation that I'm going to talk about focuses on the environment, the agriculture waste that gets generated, and how these guys are using it to generate biodegradable packaging material. So the rice stubble, which comes from the rice fields, is collected by these guys. They put it in the different bags and grow a mushroom into it. So because of the roots of that, the mycelium of the growth, it takes a shape of the mold, and they're able to convert it into a biodegradable packaging material. So with all these examples, we understand how these scientific enterprise are contributing towards the Make in India or the Atmanirbhar Bharat initiative of Government of India. So if bioentrepreneurship is a career that interests you, you can approach us. There is a very easy selection process. People can come to us with an idea, they apply, they go through a screening process, and they start incubating. And from the very next day, they can start generating proof of concept of the idea that they have. So now who can apply? As a student, a graduate, a postgraduate, or a PhD, anyone can apply in this program, and then you can come and start working here. You can start your startup as an individual, or you can make a company and start. So there are no limitations for you to start. So as a student, I would encourage, if you have an innovative idea, keep thinking about it, discuss with people, see what are the possible options around, and keep that idea with you. As soon as you are in your graduation, you can start working towards it, executing the idea that you have. And these are the various funding opportunities which exist for the entrepreneurs around. And you can see that there are a lot of options which are available by the government to fund. So funding is not an issue. And you can see one of the schemes that I would like to specifically talk about is EUR. So this scheme is specifically dedicated for students and is applicable. The eligibility for this is to be a graduate. So once you are doing your graduation, you are eligible to get such a funding. So with that, I would encourage all the young minds to think about this and the bioentrepreneurship as a career option. And in case you decide upon it, do reach out to us. This is our email and this is our website. And I would request Dr. Bhaskar to get the video for the bioincubator, please. Thank you so much.
Thank you so much. Please, yeah, I'll share the screen.
Welcome to BSc BioNest Bio Incubator, a leading bio incubator located in the National Capital Region on Gurugram Faridabad Expressway, with a mission to stimulate the establishment and growth of biotechnology-based startup companies and to translate their research findings and ideas into viable commercial entities.
Bioincubator at RCB has been established to support research and innovation in the area of biotechnology. We provide enabling environment to young entrepreneurs to convert their ideas into an enterprise.
It is funded by BIRAC, managed and operated by Regional Center for Biotechnology, which itself is an institute of national importance under auspices of UNESCO. It is an institution of education, training and research with a strong focus on capacity building. BBB provides excellent incubation facility and infrastructure spread across 35,000 square feet. The covered area of BBB includes lab spaces, office spaces, professional business suites, culture facility and instrumentation facilities.
BBB started its operations in November 2018 with an aim to support and nurture the innovators in the healthcare sector. This state-of-the-art facility provides all critical services which are required to support an innovator to move on a fast track to success.
The incubator provides globally competitive and superior incubation services to young startups and innovators and helps them to become successful enterprise. BBB can accommodate around 35 young startups in its facility.
RCB has given us the platform to establish ourselves with all the required facilities.
This facility will be very useful to all the startups for implementing their innovative ideas.
For very young startups, BBB provides shared wet lab benches, which are economical and provides them a workstation as well. On the other hand, independent lab cubicles are available for innovators who need bigger lab and office spaces. Apart from this, independent business suites are also available at a very affordable cost. BBB also has an in-house central instrument facility. This is a state-of-the-art equipment facility and caters the need of young entrepreneurs and innovators. Young entrepreneurs at BBB can also avail the facility of spacious and well-equipped meeting rooms to conduct discussions and business meetings, which are very much a part of the premises. An advanced technology platform center is also a part of the facility. This center is equipped with cutting-edge technologies to conduct experiments that will provide deep insight in biological processes and provide the best opportunity to translate these discoveries for commercialization. Various facilities such as flow cytometry, mass spectrometry, optical microscopy, electron microscopy, genomics, molecular interactions and protein purification are available at ATPC. BBB is operated and managed by seasoned and skilled professionals with rich corporate experience. The team acts as a support system for all startups at various stages of their development Some young entrepreneurs have already started their journey at BBB, and their experience has been encouraging.
BBB has a state-of-art facilities, expert mentorship.
The lab space as well as the support staff is very good.
Highly subsidized resources they offer for new startups such as ourselves.
We came here for the new ideas and start working here.
By BBB invites all young startups and innovators to come forward and experience the unique startup ecosystem of the facility. We hope our efforts will surely help in converting their dreams into reality.
Thank you, Sumanji, for such an interesting presentation. And I hope anyone who has a biotechnology-based brilliant idea can contact our bioincubator for their venture and seeking the support that we offer. This brings us to the end of this interesting scientific journey, Vigyaan Yatra into RCB. To conclude the session and close it, I invite Dr. Avinash Bajaj to take the platform, please.
Thank you very much, madam. I think with this nice presentation by all of my colleagues, we have come to end this Vigyaan Yatra, which is a part of the Science Festival ongoing. So with this, I would like to emphasize to especially the school and the college students to see the RCB website. You can be in touch with us through email, right? You can ask us for different questions. We all will be happy to reply to you and to host you here for different training programs. And with that, I would like to thank Dr. Deepika Bhaskar, Professor Sudhanshu Vrati, and all of my colleagues who have put a lot of efforts in the last 2 weeks to arrange this, make these presentations. And with that, I wish all the best to all the students who are watching this and who will be watching this when we will share this on the YouTube. Thank you very much.
Thank you, Dr. Bajaj. Thank you, everyone at RCB who's made efforts to make this possible. And thanks, everyone, for joining us. We now close this session. Good luck for pursuing a scientific career. Goodbye.